share my screen. Is it share? Okay, no, not this one. Okay, can you see properly? Is it big enough for everyone? Yeah, okay. Or do you want me to do it in slideshow? Because I want to jump from one presentation to another. But if it's really better, then yeah, maybe for this one, I'll do that. Okay. No, not that. Okay, so you're behind it. Is that okay? Just show me a thumb up if it's okay. Okay, great. So I'm going to go between two type of presentation. So that's today I would like to talk about the treatments and what we need to get a treatment and our strategy to get there. So the treatment is really the goal. That's the center of everything we do. That's the ultimate goal is really to get a treatment. But what do we need to get there? So we need to have knowledge in basic research. We need to know how we are going to do the treatments. We need to know how we are going to test the treatments. So first, not in human, because you cannot go directly in human. So we need to have the proper models and what we call assays. So that's the way to test molecules or any treatments in cellular models or in animal models. And then we need to have really well-defined assays, things that we are going to test in patients. So that's basically the tools, how to test. We need the patients, because if you are not there to participate, there is no test going on. We need the basic knowledge, and we need to know what kind of treatment we want to focus on. So that's, and thank you, Zach, because that's a very nice way to present it, and that's Zach Burns' slide. So I will go through the different type of treatments that we would like to focus on in the. I'm going to put you on the sides like that, so that I can see if you are reacting or not. So that's more a question to understand before going deep into the portfolio, what we do, understand what's where the choices and how in the whole strategy we go in the timeline. So the first thing I just told you was to develop the tools, how we are going to test anything. Then we ask the patients what's important, because that's Myhre is multisystemic, it's very variable from one patient to another. So we cannot address everything at the same time. So we had to really understand what's the most important and what are the priority for the patients. So from a survey that was conducted 2-3 years ago on 46 patients at that time, I had to, we identified that it was cardiovascular and respiratory were the biggest issues, and then behavioral. So all of those, in fact, are linked to fibrosis and stenosis. So the obvious priorities were to work on fibrosis stenosis, and the application would be cardiovascular, respiratory, and the equivalent of fibrosis in the brain to work more or on autism and intellectual disability. So what we thought would be the good way to go is the FAP CAR-T. So that's immunocell therapy that is targeting fibrosis. So that's the point, the central point. Small molecules that will tackle the problem almost at the origin. So we are looking at small molecules that will act on SMAD4. So if you remember last week, no, last month or even the month before, I talked about the Myhre syndrome. So I explained that the problem comes from a mutation in SMAD4 that gives a gain of function of SMAD4, and that triggers a lot of different issues in many systems. So that's the reason why it's called multisystemic. And the common mechanism is fibrosis and stenosis. So we decided to target the fibrosis and to target with small molecules. So small molecules is basically the traditional drug that you take in pills. Those are small molecules. And we decided to target SMAD4. So try to reduce the activity of SMAD4. So we have new chemical entities, which means that it's really new drugs. We have repurposing, so that's drugs that are on the market and that we can use also, that act also on SMAD4. And we have repositioning. It means that those are drugs that are in clinical trials, so they are not yet on the market, but they are already tested in humans. And the last part is gene editing, so really going at the origin of the disease, correcting the mistake that is in the DNA. And the last part is translational research, but then I will talk about that, I think, next month. So going back exactly to the type of treatments that we wanted to work on. So the main one is FAP-CAR-T. That's we'll go in slideshow. So what's that FAP-CAR-T? So that's really to target acute threats like tracheal stenosis, for example. And also the idea is maybe to use it chronically with a few sessions, maybe in a time periods for a month or two, and then stop for a long period. But we need to study that first. The first idea would be to use that in acute threats. So what's the principle of FAP-CAR-T? So it's CAR comes from chimeric antigen receptors. T is the type of immune cell that we are going to use, and it's a cell therapy. So basically, we use the patient's immune system to identify and kill specific cells. So for Myhre syndrome, the cells we want to kill are the fibrotic cells. So we use a recognition signal, which is a protein called fibrosis activation protein. So that's the FAP. And we are going to put that in the immune cell of the patient so that those cells can recognize which type of cells to kill. And for Myhre, that will be the fibrotic cells. One big advantage for Myhre syndrome is that they noticed that when I say they, it means the company that developed the CAR-T or the academicians, the scientists developing this technology. They realized that if you increase SMAD4 efficacy, so function, you will improve the efficacy of CAR-T treatments. So for our patients, for once, the fact to have a gain of function of SMAD4 is playing in your favor. So Myhre patient would be, in fact, theoretically should respond really well to CAR-T treatments. So we gave a grant. We invested in that technology. We gave a grant to a team of, on the one part, there is Isabelle Richards' team at the Geneton in Paris. They are generating the FAP-CAR-T. When they answer to our research proposal, they showed that they were already working and already developing FAP-CAR-T for other diseases. So that was good for us because we don't start from scratch. We don't start from nothing. There is already the project is already well advanced in the production of FAP-CAR-T. And the other part of the team from the FabCAR-T project is Valérie Cormier from the Imagine Institute. So she's the scientist, the clinician who discovered the gene linked to Myhre syndrome. So she's at the origin of the discovery of the disease. And she's the only clinician having a clinical trial now on patients, on Myhre patients. So she's also a very good fit for us for investing, giving the grant. So they are working together, the team of Valerie investigating the fibrosis part of the disease of Myhre, and Isabel Richards producing the tool, producing the FabCAR-T. So the first part of the work is really more with Valerie, who is identifying when to treat, what to treat, where's the fibrosis. So that's and they do that. Well, she sees a lot of patients, but they have also developed a mouse model, which with the 500 mutation. And they have discovered that there is a spontaneous tracheal stenosis in mice of that model. So that's a good model to test the FabCAR-T to see if it's going to work on the tracheal stenosis in particular. So they are at the same time they are investigating all the different organs, trying to monitor when fibrosis occurs, at what time during life, and they go in. They take everything, the trachea, of course, but they take the lungs, the liver, kidney, muscles, and they are going to work also on brain, but that would be a part done in the US through a company called Psychogenics. Isabel Richards, on the other hand, is producing the FabCAR-T from mice, and she's also investigating a new way to get the FabCAR-T without removing the cells from the patient, which is the traditional way that clinicians use CAR-T cells now, is that they take the CAR-T, and I will explain that. So she wants to try to invent a new way to produce the FabCAR-T than the other one. So we are talking about immune system. So just for you to understand, what's the is there any questions? I had the feeling there was a question. No? Can I go on, or I heard a ding for a question? Yeah. Okay. So the T cells, they are what we call the warriors of the immune system. They are important because they are going to recognize the cells that they have to kill with something that is called an antigen, and then they are going to kill the cell. Then they are going to attach to the cell they want to kill and send another link, and then attack the cell and kill. So when they are very alert, when they recognize this antigen, they multiply, and they become like an army, and they really hunt all those cells everywhere in the body. They are circulating, so they are really on the watch. And when they see that there is this signal, that means that the cell is really bad, then they stick to the cell and kill it. So how does it work in human, in patients? So the CAR T cells, I'm sure most of you have heard about that. They use it a lot in tumor, now in cancer. There was a safety study on I can't remember. I don't want to say. It was a really large study. I don't remember the exact number of patients they studied, but they showed that the toxicity is very low compared to other treatments. There is very low side effects, and the toxicity is really it's at a good level comparing to, of course, to the threat that we can have with tracheal stenosis or with tumor. So basically what happens is that the T cells are taken from the blood, from the patient. They are, we grow them, they are genetically modified to put the CAR that is going to recognize specific cells. So for in our syndrome, that would be the T cells would be modified to have this arm to recognize the FAP, so the fibrosis activating protein. Then we grow those cells and we re-inject the cells in the patient, and they are going to continue multiply within the patients and recognize and kill, not for us, not the cancer cells, but the fibrotic cells. Do you have any questions, or is it clear? Remarks? No, I can go on. So that's one of the project. That's our main investments. So we chose to invest in FAP-CAR-T because it is a safe treatment. This is a treatment that is already well advanced for other diseases. It's used by many clinicians. So we thought it would be the side effects are really very low. I mean, there is no treatment without side effects. There are some side effects, but very low compared to other treatments that can be less specific. So we decided to go for that one. So that's the first project that we invested in. The other project I told you was the small molecules. So I'm going to go in slash. I'm finished on FAP-CAR-T. Do you have any questions on FAP-CAR-T? Because now I'm going to talk about small molecules. No? Nothing? Oh, was I that clear? Wow. So the next one is small molecules, so really traditional drugs. The team in Spain of Maria Macias, they screened 260,000 compounds, so molecules, on how SMAD4 can form the complex with SMAD2 and 3. So they didn't have cellular models, they just had the protein and the SMAD4 protein, the other SMAD2 and 3 proteins, and they were, so it makes a complex together, so it stick together to be able to work. And they bombard it with different molecules, and they look at which molecules are going to separate them or destabilize them. From that 260 compounds, 800 compounds were shown to be able to destabilize this complex. So that's exactly what we want, because the problem with Myhre is that the SMAD4 and SMAD3 stay together too long. So it sticks together too long, and then there is a gain of function. So it works too much. So what we want is to separate them. And they found 800 compounds that could do that at the protein level. It doesn't mean that it's going to work in human, but at the protein level, they were able to identify that. Maria Macias, she's a chemist, so their job is to create new molecules and test molecules at the molecular level. But then we came to them, they came to us. Well, we are working together, and those compounds are going to be tested in the cellular model that we developed last year. So those all compounds are going to be put on patient cells. So we chose two different type of patient cells. Well, it comes from stem cells from the patient. So we take the blood from the patient, we modify the blood cells to go back to the equivalent of a stem cell, and then from those stem cells from the patient, we choose to reprogram them in vascular cells or in brain cells. Well, we can choose. So we chose those two, vascular and brain. So those compounds, those 800 molecules are going to be put in October, mid-October, are going to be put on the cells from the patient, from the two main mutations, the 500 and the R496C, and we are going to check if the let's say bad behavior of the vascular cells are going to be corrected by those compounds. Then we'll go up, select the best compounds, because we cannot go on and develop 800 compounds. We would need billions of dollars. So we are going to select the best compounds and then go to the next step, organoid if we have them at that moment, because we need more money to develop the organoids. We just have the vascularization in 3D, but we don't have really the organoids. The skin organoid and the brain organoid are not yet ready. But what we can do is go from this 3D vascular system that we have to the mouse model and go on like that up to enough characterization of those molecules to say that it's safe, that it's working on the mechanism, and that we can test them in human. So next, so I'm going to stop sharing. Do you have any questions on that? No? I'm surprised. Okay.
It's all clear, thanks.
Okay, so just something else I wanted to say on that part is that among these 800 compounds, we have new molecules, which means that it takes years and years because we have to prove that they are not toxic, that they can or cannot penetrate the blood-brain barrier. It's years of work. But on the other hand, it's not only new molecules. We have repurposing drugs. So that means that those are drugs that are already on the market, which means that all the toxicity studies have been done. So we save years of research. The administration and what we call phase 4, so really the action of these drugs on a large patient population has been done too. So that would be perfect if we can have something that act on the SMAD4 and that are repurposing drug. That would be really great. And we have also repositioning drugs that are less specific to SMAD4, but that already passed this what we call clinical trial phase 1. So all the toxicity in cells, toxicity in humans already done. So that saves also a lot of time. So what we are going to do is prioritize those molecules compared to the new molecules. And if the Institute IRB in Spain wants to develop their own new molecules, that's okay. We can still give them the opportunity to test them in the mice, but we are not going to invest 100 millions that we don't have in testing all those new molecules. So that's the second investment that we did, is to test those molecules. The third thing we work on, where is it? Oh, maybe I need to share if you. I want you to see it. No, I won't share that. So the next thing we work on. Karen, do you have a question? No. Oh, sorry. I thought so. So the next thing is repairing the gene mutation. So we have invested not that much money because we want to check first if it works or not. So we have the two results now for the two main mutation, the 500 and the I500 valin and the R496C. We were able to, nationwide, was able to show that it's possible to correct them. So they corrected them in now in also those stem cells from the patients, and they are sending the cells to our preclinical platform to check if we rescue the phenotype. So the thing we did is that you remember last time we had a meeting, I explained that the mutation is only on one of the two chromosomes that you have. Maybe I can go, don't get sick. So that's the mutation is just on one of the two chromosome 18 for Myhre syndrome. So what we do, the idea is that we trigger with CRISPR-Cas, we trigger a break exactly at the site of the mutation in one of the two, so only where there is only one chromosome that has the mutation. So exactly at the site of mutation, we create a break, and then we use the natural ability that every human has, or even yeah, that everybody has to repair the DNA. So every day you have DNA that is damaged by the UV lights, by anything, or just by replicating, it's damaged. So we have 4 or 5 different ways to repair our own DNA. So the principle that we want to use in the case of Myhre is to trigger damage at the site and then let the body repair itself. So why can't I? So the idea is to really target here, damage here where the mutation here is, or for the other one, target here, trigger the mutation, and then let the body repair. Oh, there was animation. So yeah, use the CRISPR-Cas. Look at that. Use the CRISPR. I'm not really good in animation, but use the CRISPR-Cas to really target the mutation, and then we'll use the natural DNA repair in your cell. So basically, what happens is that the other chromosome, the chromosome here, that doesn't have any mutation will come close to the DNA that the other chromosome that is now damaged with the CRISPR-Cas, and the body will use that as a template to repair. And then at the end, it's repair. I'm not going to go in the details of the DNA repair. So that's stop sharing. So that's the last one. So where we are now is that we could see in cells that we have around 88% for one of the mutation and around 80% of correction. So the question is 88% enough to rescue the phenotype? Are we going to have cells that are able to function properly? We will never get 100% of correction. I mean, nobody can do that. The next question is, what's the percentage of cells that we can have corrected at a level of 88%, depending on the delivery system, how to get this CRISPR-Cas to the cell. So that's the next step we are going to work on. Is that. And at the same time, we are checking that it's really rescuing the phenotype. So that's the third project that we really invested as a treatment. We we give money to scientists in other things, such as, maybe I can show you what we did. So share. I can share the desktop. I prefer that. So what we did last year is really create all the tools to test. So that was really the main focus. So we had the patient-derived iPSC. That's a mutation with the RR496C. That's endothelial cells creating 3D vasculars here. So that's our results. We are going to work on that. We didn't do it yet. That's organoids. So we'll start the blood vessels, the skin organoid. We'll see if we can afford to develop that. And we have developed the mouse model. We are finishing in January. The new mice model will be finished. The R496C mice will be finished, and we are going to characterize them. So that was really 2025, the whole year, well, starting August 2024 up to now. This was really we spend a lot of our energy in that. And for 2025, from October to the next year, we are going to spend more energy in the treatment, testing the treatment and developing the treatment. And at the same time, work on the clinical trial design with all the clinicians together with us.